Using this system, we can collect sub-millimeter clusters of human adipose tissue in which the whole microenvironment of regenerative cells is intact. The whole microvascular system is also intact. And so we can use these sort of microorgans for in vivo transplantation, for in vitro culture, and for cell isolation for further characterization. The main rationale for the use of this system in the research laboratory is that the process is totally enzyme-free, and so it allows the rapid and efficient processing of human adipose tissue while maintaining the microarchitecture and notably the microvasculature of the tissue totally intact. And so we can directly study a cell therapy product which has proven already very useful in the clinic.
This closed system technology is an easy-to-use technique for harvesting, processing, and re-injection refined fat tissue intraoperatively and can be successfully applied in cosmetics, orthopedics, proctology, and also gynecology. These methods allows further study of the impact of fat in tissue regeneration.
The procedure will be demonstrated by Bianca Vezzani, a postdoctoral fellow, and by Mario Gomez-Salazar, who is a PhD student.
Working under aseptic conditions, within 16 hours of harvesting,